The leaves of plants like corn, sugarcane, and crabgrass show cranch anatomy. In cranch anatomy, cuticle is present on both dorsal and ventral surface of leaf. Below the upper cuticle and lower cuticle, upper epidermis and lower epidermis are present respectively. Mesophyll cells are clustered around the bundle sheath cells. Bundle sheath cells line the veins or vascular bundles. The veins or vascular bundles contain xylem and phloem. Bundle sheath cells contain abundant chloroplasts. So, bundle sheath cells act as a site of photosynthesis and sucrose synthesis. Below the stomata, substomatal air spaces are present. Mesophyll cells are adjacent to the substomatal air spaces. Through stomata, environmental gases like CO2 and O2 go into the substomatal air spaces. Also through stomata, CO2 and O2 gases come out from the substomatal air spaces to the environment. During C4 pathway, first of all, environmental CO2 molecule passes through stomata and reaches to the substomatal air space. From substomatal air space, CO2 molecule passes into the cytosol of mesophyll cell. In mesophyll cell, a cytosolic enzyme, phosphoenol pyruvate carboxylase, then catalyzes a biochemical reaction in which cytosolic CO2 and phosphoenol pyruvate react with each other to form oxaloacetate and PI. Oxaloacetate is a 4-carbon compound. This reaction uses energy released during ATP hydrolysis. This reaction has a negative delta G. Therefore, CO2 fixation will proceed even when the CO2 concentration is low. The oxaloacetate, NADPH, and H plus ion then react with each other to form malate and NADP plus. This reaction is catalyzed by NADPH dependent malate dehydrogenase. The malate is a 4-carbon compound. In this reaction, the oxaloacetate is reduced to malate. The malate then passes through plasmodesmata and reaches to the chloroplast stroma in bundle sheath cell. The malate in the mesophyll cell moves into the bundle sheath cell by a special transporter. In chloroplast stroma, malate and NADP plus react with each other to form pyruvate, NADPH, and H plus ion with the release of CO2. This CO2 is produced in the chloroplast stroma. This reaction is catalyzed by NADP plus Melic enzyme. As in this reaction, CO2 is released. This reaction is called as decarboxylation reaction. The CO2 released from this reaction then enters standard Calvin cycle. Calvin cycle then releases triose phosphates. These triose phosphates then reversibly form starch. This starch remains in the bundle sheath cell. Triose phosphates are also converted into sucrose. From chloroplast stroma, the sucrose then moves into the cytosol of bundle sheath cell. From the cytosol, The sucrose then passes into adjacent vascular tissue. Sucrose is carried to the rest of the plant via the phloem. The pyruvate then moves into the chloroplast stroma of mesophyll cell via plasmodesmata. After reaching the chloroplast stroma of mesophyll cell, the pyruvate, ATP and PI react with each other to form phosphoenol pyruvate, AMP and PI or inorganic pyrophosphate. This reaction is catalyzed by pyruvate phosphate dikinase. In this video, molecules written in blue color are present in the cytosol and the molecules written in white color are present in the chloroplast stroma. This slide shows the structure of oxaloacetate, malate, phosphoenol pyruvate and pyruvate. C4 pathway is called as such because in this pathway CO2 fixation produces 4-carbon organic acid oxaloacetate. The other name of C4 pathway is Hatch-Slack pathway after Marshall D. Hatch and C. Roger Slack. Now some important points. Phosphoenol pyruvate carboxylase is found almost exclusively in C4 plants. Phosphoenol pyruvate carboxylase is not sensitive to O2. Bundle sheath cells do not have PS2. C4 plants are superior to C3 plants in utilizing the available CO2 because C4 enzyme phosphoenol pyruvate carboxylase has a higher affinity for CO2 than does Rubisco in the Calvin cycle. Net rates of photosynthesis for C4 grasses can be 2 or 3 times the net rates of photosynthesis for C3 grasses like rice, wheat, oats. Please like, subscribe and share.